Hi, hello everyone. Today we will talk about parthenogenesis. Firstly, we need to know what is the background of our study case. A zoologist working in a reptile conservation center discovers that some female Komodo dragon have produced offspring without mating. DNA analysis confirms that these offspring are clones of their mothers, and this phenomenon known as parthenogenesis. And we will explore how parthenogenesis works and its implication to species survival. And as you can see here, this is the topic that we will discuss and present to you guys. Now, this is our team members, and we have me, Muhammad Del Haris, and then Muhammad Ammar Baki, Muhammad Ammar bin Muhammad Mahazi, Muhammad Ammar Din, Muhammad Fahizi, and Isma Faris. Let's go to our first topic, which is what is parthenogenesis, and it will be explained by my friend Fahizi.
Let me explain a little bit about this topic, which is parthenogenesis. So, what is parthenogenesis? Parthenogenesis derives from Greek word, which translates to virgin birth. It is a type of asexual or a unisexual reproduction where offspring develop from unfertilized egg, resulting in genetically identical individuals. You may be wondering, how does parthenogenesis occur in animals? Let me explain it to you, but I will focus on Komodo dragon since it is our main topic for today. Parthenogenesis occurs when the estrous cell acts like a surrogate sperm to fertilize the main egg cell in Komodo dragons, where an unfertilized egg can still develop into an embryo. It is usually produced male because of how sex chromosome work, where it carries ZZ combination. There are also a first documented case of parthenogenesis in Komodo dragon happened in 2006 at Chester Zoo involving a female named Flora.
What are the genetic consequences of offspring produced by parthenogenesis? Parthenogenesis can result in reduced genetic variation. Offspring that produce without mixing genes from two parents can result in low diversity among the species. Next is lower adaptability. Reduced variation makes populations less able to adapt to new diseases, predators, or environmental changes. Parthenogenesis can also accumulate deleterious mutation. Without recombination, harmful mutation build up over generations. Next is short-term reproductive advantage. Population can grow quickly because no mating is required, but it can also affect long-term health of the species.
And now, let me explain about in what situation would parthenogenesis can be advantage for species. The first one is independent reproduction. Females can reproduce on their own without needing a male in isolated areas or have very few males. So, even if female is alone, the population can still continue. Secondly is rapid population growth. It allows fast reproduction because all individuals can produce offspring and useful for colonizing new habitat. This helps the species quickly fill or colonize new environment where they recently arrived. Next one, stable environment. Parthenogenesis work well in stable environment where the conditions do not change much. Since offspring are genetically similar, they can survive well if conditions stay the same. And last but not least, energy and time saving. Parthenogenesis saves energy and time because female don't need to find a male to compete for reproduction, and this allowing them to focus on producing offspring more efficiently.
What are some animals besides reptiles that can reproduce via parthenogenesis? First group is a fish. The first example is bonnet shark. They have exhibited facultative parthenogenesis. That means They can switch between sexual and asexual reproduction. The second example is Amazon molly. They reproduce via form of parthenogenesis called gynogenesis. The female needs to mate with a male from a related species to trigger its development. For the next group is a bird. First example is a California condors. They hatch from unfertilized eggs. And the second example is a domestic turkeys. It typically facultative and often result in univiable offspring or male. We continue to the third group, which is amphibians. First example is edible frog. They reproduce via hybridogenesis, a process where one of the two parental genomes is eliminated entirely from the egg-producing cell before meiosis. For the second example is unisexual mole salamanders. They reproduce process called kleptogenesis. They must mate with males from a coexisting sexual species to acquire sperm, but they use the sperm presence only to activate their egg. We continue to the last group, which is invertebrates. First example is bees. They reproduce via parthenogenesis called arachnotoky. And the last example is mites and ticks. They usually reproduce asexually.
Last but not least, I will be presenting about how parthenogenesis challenges our traditional understanding of reproduction in vertebrates. Many of us grow up learning that reproduction in animals always involve a male and a female, where sperm and the egg combine to form a new organism. However, parthenogenesis shows us that reproduction is far more complex and flexible than we once believed. Without wasting our time, let's start with the first challenge. The first way parthenogenesis challenges our understanding is by questioning the necessity of the male in reproduction. Traditionally, we believe that sperm is absolutely required to start embryonic development. This belief is the basis of how we understand sexual reproduction, which is two parents are needed, each contributing half of the genetic material. However, parthenogenesis shows that in some vertebrates, the egg can activate itself, meaning it doesn't need a sperm to begin development. Not only that, but the egg also has mechanisms that allows it to restore a full diploid chromosome count on its own. This means the offspring can develop without receiving any genetic contribution from a male. This challenges one of the most fundamental concepts in biology, that the male's role in reproduction is essential. In species that use parthenogenesis, the female alone is capable of producing the next generation. This raises bigger questioning about how reproduction evolved, and why certain species rely on sexual reproduction while others can switch between sexual and asexual strategies depending on their environment. In other words, parthenogenesis shows us that the role of the male is not universal across all vertebrates, and reproduction is much more flexible than we once thought. The second major challenge relates to genetic diversity. We usually learn that mixing the genetic material from two parents help produce variation in offspring. This variation is believed to be very important for adaptability, disease resistance, and long-term survival of a species. However, in parthenogenetic reproduction, the offspring receive genetic information only from the mother. This means the offspring are typically very genetically similar, often described as half clones of the mother. Because there is no mixing of genes, we will expect this population to struggle with adaptability. But what scientists have discovered is that many species capable of parthenogenesis can still survive and even thrive, at least in the short term. This challenges the long-standing idea that high genetic diversity is always necessary for survival. In some environments, producing offspring quickly without waiting for a mate can be a significant advantage, especially when population numbers are low. This also raises interesting interesting question about hybrid species. Some parthenogenetic species actually originate from the hybridization of two different species. These hybrids sometimes cannot reproduce sexually but can reproduce asexually through parthenogenesis. This unusual combination of phenomena, hybridity and asexual reproduction pushes our understanding of evolution and genetics even further. The third point highlight why parthenogenesis doesn't naturally occur in mammals. This absence is actually very important because it reveals a unique reproductive barrier in mammals. Mammals have a special system called genomic imprinting. This means certain genes must come specifically from the mother and others must come from the father. These imprint genes have different roles, especially in regulating growth and development. This means that mammals cannot bypass the male contribution in the same reptiles or sharks can. The fact that mammals uniquely rely on both parents shows how evolution can lead to special reproductive constraints in different groups. Therefore, the complete absence of parthenogenesis in mammals helps scientists understand the importance of parthenogenes and the evolutionary significance of imprinting. So that's the 3 points on how parthenogenesis challenges our traditional understanding of reproduction in vertebrates. That's all, thank you.
Now, from all of our explanation, we can summarize that parthenogenesis is a type of asexual, which is a form of unisexual, where offspring develop from unfertilized eggs. And we also can see there is some advantage for this parthenogenesis method, which is it for independent reproduction, and it can stable environment, and it also for rapid population growth and can save energy and time. But this parthenogenesis also can affect in reduce genetic variation, also lower adaptability, accumulation of mutation, and a short-term reproductive advantage. And also with parthenogenesis, we can understand that this is a necessity of male and is required for short-term survival. And also, mammalian don't do parthenogenesis. And this is the list of reference that we use to doing research and explain about parthenogenesis. And that's all about parthenogenesis in Komodo dragon from us. I hope you can learn something and enjoy our presentation. Bye bye.